Everything's good now?
Yeah.
Great. Yeah, thank you to the organizers. I'm really excited to be here and share my group's work. Right, so title of my talk, Mapping the Social Brain with Synapse-Level Connectomes. So I'm a new-ish group leader at the Francis Crick Institute. The group started about 2 years ago. So I'm going to talk a bit about past work and then what we're doing now with it. So generally, though, we're interested in how different animals are interacting and really on a single cell neural circuit level. So how animals recognize each other, coordinate their movements, behaviors generally. And some examples of this are, kind of pairwise interactions between animals can underlie starling murmurations, for instance, can allow animals to decide, which groups to join, which groups. So this is kind of applicable across many behaviors across the animal kingdom. Kind of the conceptual framework I work in is thinking about how do the circuits actually work. So we've seen this video before in this conference already. I really enjoyed this video though. But the main point I wanted to make with this one is that even simple stimuli will cause brain-wide functional activity. So you really need to understand, if you want to understand how the circuits work, you really need a map of the brain and some level, how the neurons fire, and then have some way of linking this to a behavioral role. And it's really hard to do all of these things, particularly if you want to go down to individual circuits. And so something has to give, and what we decided to give is we study a simple organism. So the organism we study is the fruit fly larva. And the larva is great because you can use electron microscopy to actually physically map, individual nodes, neurons in the brain, and all of the edges in that connectome. And additionally, we have this genetic toolkit that's available in fruit flies that allows us to target individual pairs of neurons in the brain, link those toolkits to the connectome, and then look at functional activity or manipulate the activity of individual nodes, again, that we know in the connectome. Okay, and so how do we actually get connectomes? This is what the larva looks like. Its head's on the left, tail's on the right. This is the central nervous system, really tiny. And that's what facilitates this electron microscopy reconstruction. So you're kind of looking at Z slices going through this brain. And that's how we actually get the circuit reconstructions. I wanted to kind of go through this a little bit just so you understand, how we do this kind of stuff. So essentially what you do is you dissect out the central nervous system, you fix and stain it for electron microscopy. And then what we did previously is we did serial sections, 50 nanometer. And then we imaged with a 4x4, roughly nanometer XY resolution. And when you do that, you get these slices. And previously, so this is my postdoc work now, we manually reconstructed each and every neuron by just going through and identifying where the different bits of ARVA were for those neurons. And when you do this across the whole neuron, you get a reconstruction that looks like this. And as you go along, you can also find these synaptic sites. So there's so this is a pre-synaptic neuron here, and these are a couple of post-synaptic partners, for instance. And if you do this densely across the entire brain, this is how you actually get the connectome. So this is kind of scrolling through those slices, and these are the manually reconstructed neurons coming out. So in this connectome, about 3,000 neurons, 500,000 synaptic sites. And this is the morphology of that. And so what you're seeing here are the two brain hemispheres, this region called the subesophageal zone, and then the ventral nerve cord. And you can kind of think of these vaguely as the equivalents of a brain stem and a spinal cord. But what's great about this connectome is, it's also linked to a lot of experimental data. So we know who all the sensory modalities are in the actual network. We have all of the connections between brain hemispheres, and we also know which neurons are controlling the behavioral output. And so this is what the actual connectivity matrix looks like. So all these little blue dots are individual synaptic connections between literally one presynaptic neuron and one postsynaptic neuron. And so I just added this bit in very quickly right before the talk, because I wanted to talk about this thing that has come up during this conference. Actually, people have been asking me about this. So how stereotyped are nodes in this connectome? And one thing we see that's quite interesting is that you actually have a mirror copy of each node across the left and right hemisphere. And this is known based on the connectome here, but also based on loads of experimental data. And additionally, these individual nodes can also be found across individuals, which is great because you can really compare connectomes in a really, really fine scale way, and also do experiments on those individual nodes to see what they're doing in the connectome. And then another question is, how stereotyped are the edges across animals? And to get at this, I want to talk about, two types of edges, basically just weak edges and strong edges. And what do I actually mean by that? What I mean by that in our context is, if you look at neuron A communicating with neuron B, a weak edge might have one or two physical synaptic sites between those neurons, where a strong edge will have, 5 physical synaptic sites that are actually mapped in the connectome or more, for instance. And basically what we see is that the weak edges are not stereotyped across brain hemispheres, even in the same animal or across animals, where these strong edges are stereotyped, very much so, across brain hemispheres and across animals. And this is also seen in CL again, so weak connections are variable across individuals. This is just kind of a baseline for these nanoscale nodes. Okay, and so we have all connections across brain hemispheres, so all the ipsilateral neurons, for instance, bilateral neurons sending outputs to both brain hemispheres, and the contralateral ones that are going to the opposite brain hemisphere. And by looking at all these connections, we can actually follow signals throughout all the cell types in the brain, from the periphery to the descending output that kind of produce the behavioral instruction to the nerve cord. And so you can follow these in a feed forward kind of way or in a feedback direction. Okay, so that's that. If you want more information, there's loads of analysis in the science paper, but I want to kind of move on to what are we actually doing with this connectome now. And the primary goal is to link this connectome to social behaviors. And we're doing this in 2 ways. One way is we're generating new connectomes, where we've perturbed the connectome in some way and so modified the edges in some way, and see how that's disrupting the behavior. And then the other way is we start from the behavior experimentally, and then we activate or inactivate individual nodes of the connectome and see how it disrupts different behavioral aspects of the behavior we're looking at. Now, what is the actual behavior we're looking at? Larvae do some very simple things, sometimes quite boring in some ways. They move forward, they move backwards, they can turn, this kind of stuff. But they can combine these movements into things that are much more complicated. And this is the actual social behavior we're working on. So the behavior we're working on is this cooperative foraging behavior. What you're looking at here is a fruit fly vial with food. And what happens is the animals coordinate their movements to dig these pits together, and they can access nutrients they couldn't access on their own by synchronizing their movements. A little hard to see there, but this is what it looks like kind of in the lab. So you can see that they form these clusters, and there's a lot of decision making that goes into this, which clusters join, when to leave clusters, and they synchronize these movements to dig these little pits. And what you're looking at here, this is a transparent food media on the top, and this is air, on the bottom, and this is air on the top. Okay, and so why are they doing this? I've probably mentioned this to some of you, that kind of the goal of the larva is just to eat as much food as possible so it can become an adult fly. That's the only drive they have going on, and to avoid predators. That's really it. And so what they do is they stick their heads directly in food and just eat. And they have this really funny anatomical quirk which allows them to eat more effectively, which is they can actually breathe through their tails. And this allows them to just sit in the food and never stop eating. They can just eat and breathe and eat and breathe. And then when they've eaten enough, they can go through metamorphosis and become a fly. And that's all great. And this is what they do when you just put them in food. And so this is food with some level of nutrient quality. And they just eat on their own. They don't really form these groups. But at some point, they'll eat the nutrients out of that top layer of food. And now they have to make a decision. They can dive deeper into the food and eat more, but then the hole that they burrow collapses behind them and they can't breathe. So if they choose, do they want to breathe or do they want to eat? And they can dive down and eat and come up for air, but it's really inefficient. So they're kind of going back and forth between air and food and air and food. But eventually they learn after a few days of this that they can actually synchronize their movements, dig these really deep pits that are wide and don't collapse, and then they can access air and food at the same time. And it's a really interesting behavior because we know it's learned, because if you socially isolate the animals so they don't have social experience before, they never learn how to do this. Additionally, the animals appear to they're more likely to form these social groups with kin as opposed to non-kin. So it suggests it's actually a surprisingly complex social behavior. So this is what we study. And basically our questions are what circuits and computations underlie these group behaviors, and then how are they disrupted by environmental or genetic perturbations. Okay, and so this is kind of the summary of the tools we have. So we have the connectome, the ability to manipulate nodes and edges. And the first bit I'm going to tell you about is how we're using behavioral recording to get at this. So this is the first section. Okay, so just to summarize, so this is a fruit fly vial. These are larvae in there. This is kind of how we think it happens. The larvae are first attracted to each other. They then initialize these cooperative groups, and then eventually there's some kind of group dynamics where animals choose which group they'd like to join. And so I'm going to focus on them actually forming the groups first. So this is kind of our strategy of figuring out which bits of the connectome are important for this behavior. So what we do is we take our driver line library, we have it linked to the connectome, and we inactivate neurons one by one, and then see which of these neurons has an effect on these digging groups. And any one that affects a digging group must be important for the behavior, is the thought. Okay, so to do this, we actually have built a imaging rig where we have a camera mounted at the top of the vial. And this is what it looks like. It's controlled by a Raspberry Pi, one of these kind of microcomputers. They're just a few centimeters long. And so when you're looking down into a vial, you don't actually see the animals per se. Instead, what you see is the food. And then you see the holes that result as a byproduct of this cooperative behavior. So this is a time lapse taken over a few days, and each frame is 5 minutes. So you won't actually see animals. But at the third instar larval stage, this is when they start doing this cooperative behavior, and you start seeing loads of these holes forming. And so this is what we're interested in. We're interested in quantifying these holes, essentially, and looking at them over time and their dynamics. With the thought that if you do this and look at control animals and then compare when you inactivate some kind of neuron, if you see a shift, then that neuron must be important for the behavior. Okay, so I'm going to show you some hardware just because this is what we've been doing lately. But we built this this incubator that has arrays of Raspberry Pis, so hundreds of them. They run these camera cables into the incubator. They then record from many of these vials simultaneously, so we can do about 300 experiments. And now we just have loads and loads of images like this. But then we needed to figure out how to quantify them. So we tried various kind of classical computer vision techniques, but they all failed for the most part, and we ended up just using machine learning. So we have a convolutional neural net, we fed it some training data, and now we can identify and quantify these holes. Okay, and the plan is we're going to be inactivating many types of neurons. So first sensory neurons to figure out which sensory modalities are important for the behavior. Later on, brain interneurons, as well as some neuromodulators. So that's kind of the plan for this thing. We do have the first bit of this screen completed for the most part, looking at the sensory neurons. So this is just fold change where one is kind of the control level, we just normalize there. So this is whole counts. And this is an example of some neurons involved with vision. There's some effect, but it's not significant. And then here are all the different lines we tested. And we see that when you inactivate some sensory modalities, you see decreases in the number of holes. And when you inactivate other modalities, you actually see increases. And so based on this, this is preliminary at this point, but we think that mechanosensation is important for this behavior, which makes sense. The animals are kind of rubbing up against each other. It's probably a primary way they sense each other when they're nearby, probably also how they synchronize their movements. But olfaction is also important, as well as gustation. And then importantly, the last one is proprioception. So the animals have to be aware of where their different body parts are in order to do this properly, which kind of makes sense, I think. But you know, since we have this machine learning model that allows us to quantify this, we can look at many more things than just the number of holes. So we also look at sorry, I messed up that slide. Well, I see what's happening. I think it's going backwards. Here we are. Sorry about that. We also can look at the size of holes, the kind of lifetime of holes, and other dynamics of the hole. So here you're looking at p-values, and we've compared against control. So there's many different things we're looking at now, and that's kind of an ongoing bit. But But what's kind of a shame, though, is that we can't actually see the animals, right? That would make it much more interesting if you can really see the animals. So the plan is we're taking these hits by using the screen, and any of them that look interesting, we then move on and look at group dynamics. And to do this, what we've found is that you can actually mount the animals between two pieces of glass, and just microscope slides, put a thin layer of food in between, and they'll fortunately still form these cooperative digging groups in that orientation. And this is how you can actually see the animals. So we've built another behavioral rig where we record them like this. It started out very simply, which is secondhand iPhones, actually. But it in principle worked. And then based on that, we've been upgrading to kind of more professional behavioral rigs. And this is the final version. So you can see the digging clusters there, and then you can actually see individual animals and how they're kind of bouncing between groups and deciding which group to join. And this is great, you know, if you fast forward these videos, you can really see loads of dynamics between these groups. So we're really interested in how the animals are deciding which group to join, when to leave. Kind of our running hypothesis is that each animal is making a cost-benefit decision about each group, whether they're putting in more effort than they're getting out of it, and that makes them choose to leave or stay. So these are the kind of questions we're interested in. We've built loads of these behavioral rigs at this point, so we have loads of data, but again, we had this problem of how do we actually analyze and track these animals. And so we turned to this deep learning method called SLEEP, where you annotate animals and then build a model that can then predict the different body parts of the animals, and then you kind of fine tune and go in this loop until you get nice predictions. And so we've done this for loads and loads of animals. One of my students did this. And we now have a pretty good model that can track the animals at this point. And using this kind of data, you can do things like identify the clusters in an automated way, and also identify animals that are non-clustering, for instance. So we did this using dbScan. And so using this data, what we're now doing is looking at, in the first instance, just a simple metric, so which fraction of animals are in these cooperative digging groups versus which are not. But we're kind of working now on figuring out more complex ways of actually modeling the animal behavior as a network itself. So I really like this idea of you have the connectome, which is obviously a network, and then you have the interactions between many animals. So it's kind of a network within a network model. Okay, great. And just as an example of what we've been doing with this is we're taking these inactivation hits from our hole count screen, and then we had this question of, okay, when we actually see a decrease in the holes, is it actually affecting this clustering behavior where we see the animals? And so this is an example of that. So this is just a control experiment. And here is the inactivation of the proprioceptors in the animals. And you can see it has quite a striking effect on them. I also have an example of when you disrupt the gustation in these animals. And you can see that both of these, they're quite disorganized and not really forming these nice clusters anymore. And this is just one example of quantification of that. So this is a fraction of animals within a cluster, and control, you usually have about 50% of animals, say, up here. We have two controls here because there's just some genetics that's not very important for this talk. But you can see there's a huge decrease in the amount of clusters. So it seems like that the inactivation screen provides robust hits that kind of work across different behavioral assays. Okay, but what's really cool about this is we can now take those genetic lines that we use to inactivate neurons. We can look at their morphology, and then we can match that to morphology in the connectome. And then once we have that link between the experimental data and that node in the connectome, we can then pull a connectivity list and find other neurons, for instance, and then really look at the upstream and downstream partners, for instance. And this will really allow us to form a tight link between these different aspects of behavior and the connectome. And so that's kind of the future of this project, to try linking those things. And then we're hoping, in future, once we've done this across all of these genetic lines that we have, we can then answer questions about where are the circuits involved in the social behavior located. Are they distributed, or are they localized within some particular portion of the brain? And then what kind of circuit motifs are they and how do they work? And then we'll really start getting into the network analysis kind of stuff at that point. Okay, so summary of this bit. Basically, we've made a pipeline for a high throughput behavioral analysis while inactivating individual nodes in a connectome. And we know now that olfaction, gustation, mechanosensation, and proprioception are important first instance. I think it will become much more interesting as we get deeper into the brain and really discover new circuit motifs there. This is just kind of the start. And we also, as I mentioned, have a method to link this behavioral data to the connectome. Okay, and now the next bit, I mentioned at the beginning that we're also interested in generating new connectomes and looking at when you perturb the connectome, how does this affect behavior? And so the first project in this direction, we're looking at how early social experience affects brain wiring. And so what we're doing is we have either group housed or isolated animals. And then the question is, what effect does that have on this grouping behavior? And so, so one of my students has been working on this, and basically what we do is we group house the animals for 4 days. The larvae live usually about 6 days until they go through metamorphosis and then become adult flies. So this is fairly late in development. But if you group house, as you've seen, this is a 2.5-minute video. You can see that they form these nice clusters. But if you socially isolate them so that they've never seen another larvae until the moment when you assay them, it's drastically different. So this is what it looks like here. So you can see there's not these organized groups. The animals are kind of repulsed from each other and this kind of thing. So it's very different. We've quantified that using the deep learning method I mentioned before, and you can see a striking difference between the group housed and isolated animals. Additionally, there's now been a paper about this where another group, they actually took socially isolated animals and transplanted them into control digging groups. And even in that kind of best case scenario where all of the other partners knew what to do, the socially isolated animal couldn't coordinate its movements properly and actually disrupted the group, suggesting there's something really going on with those isolated animals. Additionally, the same group found that using light microscopy, not connectomics, but they found significant changes in the axon boutons of some visual neurons, suggesting there's actually wiring changes going on. So that's kind of what we're interested in doing. So actually mapping the brains of these two different conditions using connectomics and then trying to find out what's different about them. And so to do that, we're now using an enhanced FibSim technology. So this is focused ion beam scanning electron microscopy. This is about an order of magnitude faster than the PEM that I showed you before. So you can image an entire brain in about 5 weeks, where before it took about a year of imaging time. So significantly reduced timescale here. Also, instead of manual reconstruction, we're now implementing automated techniques. So this is a pipeline called local shape descriptors, where there's a couple different 3D UNets that interact with each other to pull out different features about the shapes of neurons in this raw data, as you can see up here. And this allows you to actually segment out the neurons in 3 dimensions. So this is what we're doing now. And this is some of our data that we acquired, a test set of data. And you can see what I'll do is I'll slice through the Z slices, which are 8 nanometer here. And you'll see, you can kind of follow the neuron morphologies. And then the first thing it does is actually identify the borders between neurons and then actually segments them out. So this technology is working quite good. And it's very impressive because we don't even have training data. So the collaborator, Samia Mohinta, who worked on this with me, she's actually using transfer learning to take publicly available training data sets and using GANs to then mimic the look of our data. And it's working quite nicely. And based on this, if you go over a huge range of Z slices, you can actually reconstruct the entire neuron. So I have, here's some examples of that. So this is the raw data here, but here are actually some of the neuron reconstructions. And as an example, these are about a dozen neurons or so that are in one of these preliminary data sets we've generated here. And so what you're looking at are, the axons are over here on the left, and then many dendrites are here on the right. Okay, great. So the idea is we're kind of actively acquiring these data sets right now, and we're hoping to see, what circuitry changes happen. Our hypothesis is that we'll see differences in edge strengths, but there could be other differences. There could be new connections that don't exist yet, or even changes in some of the development of these nodes, for instance. Okay. However, the one thing we're missing here that wasn't in my previous work, and I haven't really mentioned, is that when you acquire a connectome, you actually don't know which neurons are excitatory and which are inhibitory. That's a major downside. But we've been working on this because obviously, if a neuron's excitatory or inhibitory, it completely changes what it's doing in the local circuit. So this is really essential information. So what we've done, I've worked with a software engineer, my institute, to identify, well, I should take a step back. We have experimental data that has identified which neurons express different neurotransmitters. And we have that linked to the connectome using these kind of genetic tools that I mentioned before. So we have some known ground truth synapses in the connectome. And we have a huge set of unknown ones. So the idea is that we could train a 3D convolutional neural net to then predict the neurotransmitter expression in all of these unknown synapses. And then using this information, we could figure out which ones are excitatory and which ones are inhibitory. So this is what we've done. This is kind of preliminary work from a new postdoc in the lab. And initially, this is a confusion matrix where on the one axis, you have the true labels. So we have a held back data set. This is kind of the test data set. And on the other axis, the predicted labels. And you can see per synapse, it's actually not that good. So it's only about 30 or 63, 64% accurate. However, there is a thing in the fruit fly, which is if you know for sure the identity of any particular synapse in that neuron, that is going to be the expression profile across the neuron. This is an established thing in the experimental literature. And so what we can do is we can actually take a majority vote based on all of the predicted synapses across the neuron, and whichever is the most predicted synapse, you can assume is kind of the identity of the neuron. And if you now do that and then compare to held out sets of neurons instead of synapses, it actually becomes quite effective. So now we're in for acetylcholine, 87% accurate. GABA is still about 75%, but glutamate was quite impressive, 100% accurate in these predictions. So we're now using this data to reassess circuit motifs we've looked at in the past. Okay, right, and now you can actually make predictions about all of the neurons across the brain, for instance, and then use this to run different simulations. So I didn't really get into this, but there's some techniques from the science paper where we looked at how signals propagate across the whole network. We now have that implemented with excitatory and inhibitory information. Okay. Right, so this was kind of my flash talk slide before, but I just wanted to, it's a nice overview anyway. So we have two sides to the lab. One side is experimental, where we're building hardware to record behavior, and we're tracking the animals to actually better characterize and understand their behavior. And we're also starting to develop 2-photon imaging techniques where we can actually track the animals as they're intact and freely behaving. On the other side of the lab, we have the computational group that actually takes electron microscopy data, segments it, and then generates connectomes, then analyzes those connectomes. And we have another project that I haven't really had a chance to talk about where we're looking at gene expression and how gene expression changes happen due to social isolation. And we have kind of the broad goal is to figure out how those gene expression changes actually change the circuitry and disrupt the behavior, and whether we can kind of revert some of those changes. And then kind of a summary and kind of outlook of this is what I say to the funders, for instance, is that we're using artificial neural nets to better understand the connectome, and in our case, social interactions. But we're hoping that one day we can also use these to inspire new architectures for artificial intelligence, or potentially also new ways of having models interact. So this is kind of a new thing. We have multiple different models doing things. In fact, that's one of the ways that we actually reconstruct the neurons. We have multiple models interacting. Okay, so that's all I have for you today. Thanks for your attention. I just want to thank the people that did the work. This is the experimental side of the group whose work I presented today. This is the computational side, and then other lab members. And I'd like to thank the collaborators.